Good morning, everyone, and welcome to our Minnesota Stormwater Research Spotlights, part of our Minnesota Stormwater Seminar Series. These series are made possible through the Minnesota Stormwater Research Council, which is funded in part through the Minnesota Clean Water Land and Legacy Amendment. My name is John Bilotta, and I'll be the moderator today for our two presentations. Before I get started, I'd like to acknowledge my two colleagues, Dr. Andy Erickson and John Gulliver, and our whole team over at St. Anthony Falls Laboratory for making this seminar series possible and our great collaborative work to bring these professional development opportunities to us all. I'd like to take this time to also encourage you to check out the Minnesota Stormwater Research Council and all of the projects that we are funding and completing through the Research Council. We currently have 19 different research projects being worked on to help us better understand stormwater management and increase our efficiency and effectiveness of stormwater practices and policies. So please check out the website, and while you're there, I would encourage you to check out our new 4-page highlight of our work over the last year. This is a quick read, providing you a quick glance of all the different research projects and the different research teams working on those. So check it out, pass that along to your colleagues. So with that, then I'm going to move us along into our first presentation today. I'll invite Todd for you to start sharing your screen as I do a short introduction of our first research project. And that is the pollutant removal and maintenance assessment of underground filtration systems. The purpose of this study, funded by the Minnesota Stormwater Research Council, was to look at stormwater management effectiveness of underground sand filters here in the Twin Cities metro area. These types of sand filters don't always have clear access, and we recognize that we needed additional guidance for ourselves, including future updates for the Minnesota Stormwater Manual. So this research project was funded to look at these issues, and it's being led by Todd Shoemaker and Allie Stone, both of Wenck Engineering and the University of Minnesota. First, a little bit about Todd. He has more than 20 years experience in water resource management and engineering, and he is joined by Allie Stone, who is a student here at the University of Minnesota, pursuing a degree in civil engineering with an emphasis in water resources. So with no further ado, I will hand control, and we look forward to our words of wisdom from Todd and Allie about this research project.
Hey, welcome everybody. Andy, John, Research Council, thank you for the opportunity, the funding and moving us forward on this research study. We appreciate that. We appreciate the opportunity here to share our study results and some possible next steps. So with that, we'll jump into it and describe what we've done and what we've found. So I think many of us are probably very familiar with surface sand filters. They've been around for decades. I think their design and the science of them is pretty well understood. In terms of stormwater management, I would say that they're most commonly used for sites that have poor draining, poor infiltrating soils, or on sites where there's contamination. The idea being is that you want to use filtration with sand being the media, or sand, compost, topsoil, some sort of mixture. You want to use filtration so as to capture that treated water and move it downstream rather than trying to force it through poorly infiltrating soils or into contaminated soil. The catch here, though, is that to be deemed effective, these filters, if you think about it, they have to clog. Once they clog, we know that they're effective. They're capturing the pollutants. That's all well and good in suburban areas or new developing areas, but what do you do in a landscape like this? This is, redevelopment city right here. it is existing 80 to 100% impervious, and after redevelopment, it's going to be pretty darn similar to that level of impervious. So what engineers and designers have done is that they've taken those principles from surface sand filters, and they've applied those to basically the underground environment. I'm sure many of us are familiar with these systems. You know, we're building large tanks underground with a layer of sand below it, and then a drain tile below that sand to filter the water and then convey it downstream. But getting back to the whole principle of the filter is that it is designed, we want it to clog. And as we as engineers and designers move away from a site, or we move on to other projects, the owners are left essentially with a fairly expensive system below ground that's out of sight and out of mind. So this last bullet here is just a caveat to everybody here, is just to be very clear. Our intent with this study is to evaluate effectiveness of these underground systems. We are not here to point fingers at any engineers, designers, owners, vendors, manufacturers, and even site owners. As we move through this, you're going to see that our sites are very generically named, and that is intentional so as to maintain the site and the owner's anonymity. We appreciate their cooperation in allowing us to monitor, and we didn't want to get them in any trouble. Not that we expect there to be any, but that's how we proposed it to the site owners. So with that, I wanted to show you just a generic system detail of what many of these systems look like. So in the middle of the screen here, you can see this generic detail that has two large pipes. You know, these two round pipes in the middle, from what I've seen, can be anywhere from 24 inches in diameter to up to 96 inches in diameter. So fairly standard size all the way up to extremely large. Typically, they're surrounded by a clean washed stone that the pipes are perforated. So both there's storage for stormwater within the pipes and the rock surrounding it. Now, this detail, this example, it just shows pipes, but there are other systems that are available, such as vaults, concrete vaults, or plastic domes. And a couple of our, I guess one of our examples uses the plastic dome. The other three are pipe scenarios. But below this storage layer, so the pipe and the rock, or the dome and rock combination, below that is the filtration media. Typically, that is sand. And embedded within that sand is our drain tiles. And that's where the water moves through the sand into the drain tile, and then the drain tile is out. So this volume is what we call the filtration volume. That's below the outlet or the overflow. That is typically sized based on the local regulatory requirement. That, you know, in the metro, maybe that's 0.55 inches for a linear project, 1.1, 1.25. There's some variations on that, maybe up to even 1.8 or 2 inches. So that's the local regulation typically dictates what that filtration volume should be. That volume then, that needs to move through the filtration media typically within 48 hours. So that 48-hour time frame, that is what dictates the distance or the depth between the outlet overflow and the top of that filtration media layer. As water moves through that sand, then it goes to the drain tile and on downstream. So our study goals were to assess the performance of underground filtration systems. We did that by evaluating the pollutant removal within these systems. So TSS, TP, total dissolved phosphorus, orthophosphate, and E. coli. We also installed pressure transducers to measure the drawdown time or the filtration rate through that sand media. And then finally, we reached out to the owners to determine has the system been maintained, or if it hasn't, has that lack of maintenance affected the performance? So in terms of our methods, we chose 4 sites. The sites were, I think, mainly chosen for proximity. They are in the Twin Cities between basically Minneapolis, St. Paul, north of the Mississippi River, north to Shoreview. So they're chosen for proximity. We had some options to go a little bit beyond the 494, 694 loop, and that would have been very difficult for just in terms of the budget and the time allowed for us to mobilize, to get grab samples and effectively monitor those systems. So for each of our 4 sites, we had a pressure transducer installed to measure water level. As storm events occurred, then we used grab samples to measure the pollutant removal at the inlets and the outlets of these systems. In total, we captured 6 storm events in 2020. Next few slides here, I'm going to give you a summary of the 4 different sites that we looked at. This one we're calling the parking lot site. This is our oldest site of which of the 4 that we studied. So it was constructed in 2012, so it's been online and operating for about 8 years. The existing soil is mapped by the soil survey as urban land. We did not have access to soil borings if they were conducted. So my guess here is if you see to the right of the aerial photo, there's a wetland that's located adjacent to this system. My guess is that the low or the poor draining soil within that wetland probably extends over underneath our system that we studied. So that's why filtration was chosen for this site. The light blue color there shows the drainage area to our system. The yellow arrow shows that there's 1 inlet to this system. The green rectangle is our system, and that is a plastic dome. So there's a storage rock that surrounds the plastic dome, and then a filtration layer and drain tile below that. The plans that we reviewed for this did not specifically indicate any level of pre-treatment for this site. I am familiar with the design here of the plastic dome, and I would expect that there would have been a sedimentation row to provide pre-treatment. However, there was not specific documentation to say that that was present on this site, but it's likely that it is there. And then I think the last thing I'll point out is just that filtration media is clean washed construction sand, and that corresponds to what is recommended in the Minnesota Stormwater Manual. Our second site is what we're calling the school site, and appropriately there is a school building on it. The portion of the roof, the portion of the building that is within this watershed, that was added, or that was an addition to the existing school building. So that addition is plumbed, is directed to the underground system. So here we have about 1.7 acre drainage area, and you can see the splits there in terms of it's 66% impervious, but a pretty small portion of that is that parking lot. It's mostly playground and roof. This system had 2 inlets. You can see there by the 2 yellow arrows. At each of those inlets, there is a sump manhole, and each of those sumps had a snout device on the outlet. So that snout device is intended to retain floatable material in the sump manhole and also prevent some scour of accumulated sediment to some degree. This site, There were soil borings available, and those borings indicated sandy, lean clay. So that's why filtration was chosen for this particular site. This differs from our parking lot site because this is a site with round perforated pipe. And it also uses clean wash construction sand with geotextile, geotextile layer just below that, the sand and the drain tile. The previous site that I mentioned, the parking lot site, note that I didn't say that. That was just the filtration media right on top of the in situ soil. Site number 3 is what we call our office site. One thing I forgot here, this is our newest site. So parking lot was oldest, school is newest, constructed in 2018. Office site constructed in 2016. The web soil survey indicates A and B soils on this site. Now I know that is not perfect, but that was the best information we had to look at. Soil borings were not available, so perhaps there was a contamination issue on this site. That's why they went with filtration. Pretreatment here is provided by upstream BMPs. So kind of interesting, or also a cool design on this site. There's a tree trench, permeable pavement, and a bioretention basin that treat stormwater before, or those 3 devices ultimately drain into this underground filtration system. Now that was, I kind of questioned that as I reviewed it, but this particular system, it still had the drain tile and the outlet was still raised above the invert of the storage pipe. So the designer, the requirement, whatever it was, there was still, it appeared to me that there was still some intent to provide some water quality treatment within this system. The bottom row here on our system description, you'll see that the media here is different. It's an angular wash stone, and there's an impermeable liner below the sand and the drain tile. The last of the 4 sites is what we're calling the animal site. Constructed in 2017. Soils were urban. I don't know much more than that. We didn't have any more information on why specifically filtration was chosen here. The middle arrow comes directly in from the roof, so there's no pre-treatment from that arrow. And then the inlets, so the yellow arrows on the left and the right, those 2 inlets each have 4 and 5 foot sump as pre-treatment. Drainage area is 43% impervious, 33% roof, 10% pavement. And of that roof, what's interesting here, I guess I'll reveal the name for the animal site, or why the animal site. The roof on the top right there was labeled as animal barn. So you'll see some supporting information for that later. That's why we called it the animal site. So here we have perforated pipe again, clean wash construction sand as our filter media, along with a liner below the sand and the drain tile. So let's talk a little bit about our results, our storm events. So as our target over the summer was 10 storm events, as probably many of you know, collecting samples using or going the grab sample route is an imperfect science indeed. So unfortunately, we only got to 6 storm events, but we were, I was fairly happy with our rainfall depths. You see, we got a pretty decent spread there, along with 1 0.9-inch event, which again, I don't know the rationale behind the design for many of these sites, but my guess is that 0.9 inches is probably pretty close to what they're what the design goal was for treating that depth of a rainfall within these sites. Our results will be presented in box plots for both pollutant removal and the filtration rates. So we got a lot of information to present on this slide. I'm going to take another drink of water here just to give everybody a few more seconds to digest this. I'll start on the bottom. We have our 4 sites listed along the x-axis there on the bottom. Inlets and outlets are plotted. The blue box plots represent our inlet, our influent concentrations for TSS. The gray are the effluent concentrations. We've listed the percent reductions and increases at each site, but I think many people are very familiar with the percent reduction or that concept, but I don't know as if it's fair to focus on that at this point. And we'll get into that as we continue reviewing the results. Well, I'll get to it right now, I guess. I mean, you can see from the effluent concentrations, in general, they range from what, like 3 milligrams per liter up to 10 milligrams per liter. The box in the top right of the slide gives you some context as to what's reported in the Minnesota Stormwater Manual. So in the manual, that indicates that we can expect a 75 to 90% TSS reduction and an effluent concentration 5 to 16 milligrams per liter. Based on that effluent concentration, it shows all 4 filters are doing a pretty good job. The first site, the parking lot site, shows the most significant reduction, 74%. So we're pretty darn close. We're in the ballpark to what's reported in the manual. The middle 2 sites there, school and office, are a little bit concerning because that's showing some, I guess, leaching, some transfer of sediment through the outlet. The y-axis is circled though. You can see that those influent concentrations are quite low. I believe also in the manual that the EMC values for TSS can be anywhere from 50 to 90 milligrams per liter. So the parking lot site is getting close to that, but the other 3 sites were quite low. So that's why, since our influent is a little bit below what we'd expect, that's why the reading too much into the percent reduction or percent increase right now can be a little bit misleading. Let's look at total phosphorus. I have notes up towards the top there for context. So MSM is Minnesota Stormwater Manual. So again, our influent concentrations are well below what we expect. We didn't see the level of reduction that we expected with phosphorus, but also the effluent concentrations that we measured are within what is indicated as acceptable or expected from the Stormwater Manual. So it's both kind of good news and bad news that the effluent concentrations are within that acceptable range, but it appears that there is some leaching or some sourcing of phosphorus from the filters. We also looked at total dissolved and then orthophosphate. We'll go to total dissolved phosphorus first. Both of these constituents, we would not expect any level of removal or reduction from sand filters. This dissolved material should pass through the voids within the sand. Similar to the previous 2 pollutants though, our influent concentrations and effluent are very low. So yes, we're seeing some pretty significant percentage increases, but probably not all that concerning in terms of downstream resources because that concentration is so low. Orthophosphate, similar story as to total dissolved. Perhaps if you're just looking at percentages, concerning, but the concentrations are not. It's concerning that there could be increases moving through the filters, but the we need to do more to understand exactly when our samples were taken and dive into the data a little bit more. I'll get into that here in a few slides. Our last constituent here is E. coli, and Stormwater Manual reports that sand filters should remove about 50% or reduce concentrations by about 50%. Also added in there the surface water quality standard, that's 126, I believe. And I'll point your attention to the right, to the animal site. You can see that we happen to, I think, probably accurately capture or prove that that is indeed an animal barn, given the inlet concentrations that we recorded over there. I think generally all of these sites, except for the parking lot site, really they were well below what we, or at least that standard, and there was not the necessarily the sourcing or the leaching or growth of E. coli within the underground system. Let's look at filtration rates. So we have two of the sites here. We have the parking lot site, which, if you recall, that was constructed, that's been online since 2012. And the animal site, which has been on site since 2017. Parking lot is at about 1 inch per hour filtration. So that's the rate at which water is moving through that filter media, moving through the sand. The animal site is quite a bit higher, 3 to 4 or 5 times higher, 3 to 5 inches per hour. It's possible here that that difference is solely because of the age of these systems, that the parking lot site has settled in, there's been some consolidation, there has been some accumulation of material that has reduced that filtration rate. The animal site is newer, so it just hasn't had that amount of time, that amount of material to accumulate and reduce that filtration rate. You'll probably notice the two sites are missing from this figure, though. That is one of them. Well, they're both missing. I'll say they're both missing because our pressure transducers never recorded an increase in the water level. They were always the data bounced around a couple hundredths of a foot, such that the water was never accumulating in those systems. If you recall back to the office site, that's the one that had the 3/4 to 2 inch rock as the filter medium. You can probably imagine that makes sense, that material is more porous, that it's going to allow the water to move through that much quicker than what a sand media would. So that one I think is explainable. The school site, though, that if you recall, that was also our newest site. So that's been online for 2 years. 2020 would have been the third year. My thought there is that I think as water runs into that sand layer is just acting like a sponge. That water is getting into that sand layer, and that is actually acting as the storage for runoff. And the drain tile below it is, there's some rate reduction there, but that sand is still not consolidated enough, or there's enough void space in there that the water never popped, never increased into the pipe storage that's where our pressure transducer was located. That bore out as well when we looked at the I guess we did some calculations in terms of the drainage area and the storage in that sand layer. So let me be easiest if I go back here. To our school site. So this is 1.7 acres, 66% impervious. If you do the math, the volume of runoff coming from the watershed here is about 5,000 cubic feet or so. The storage within the sand layer here is about 4,000 cubic feet. So there's a chance that these systems initially, that there's that additional storage that's provided in the sand. Now, I think in terms of design, it's probably not reasonable to take that storage into account as we design these systems, because again, if you think back to the beginning slides, our intent here is to clog these filters. We want material to build up in the sand. So I don't think we should be depending on that to store water and slow it down. So let's kind of bring this all around to some conclusions here. We'll start with the good news. So as I've noted, the effluent concentrations compare favorably with Minnesota Stormwater Manual. That's great. E. coli exceeded the MSM value, and there's no evidence of growth within the media. In terms of, I guess, my own design and advising clients or other designers, we like to default to the 1 inch per hour design filtration rate. That seemed to be proven or corroborated with the parking lot site. The animal site was much faster, but I think that is, I would say the 1 inch per hour is still a good place to start. The question mark is there just because of the rate, the fast rate at which water was moving through the sand at the school site. I explained why we think that is, and we can, if we continue monitoring that, we can see if that rate, if we eventually see some bounce in the water accumulating in that system. In terms of maintenance, our oldest site, again, that was the parking lot site, 2012, that showed the highest TSS removal. So that's great. Our oldest site is still performing. With all 4 sites, at all of our visits, we didn't observe any significant maintenance issues. There were the pretreatment devices. There was material, trash, debris accumulating. They were functioning. And I know for sure that at least 2 of the sites are receiving regular maintenance inspections. The owners are aware of these systems and at least monitoring them to some degree. Finally, we found no evidence of filters clogging. But there are some reasons for concern. So first of all, just reason for concern is just our own study here was limited, and I'm sure you probably observed that or thought that as we've described this here. We have 4 sites, 6 storms, low influent concentrations. So we are very cautious to making concrete conclusions and recommendations from this limited data. That being said, pollutant reduction for TSS and TP was lower than expected. There was some leaching, and we talked about the influent concentrations. What has been presented, or what was presented in those box plots, was all of our data, all 6 storms. What we want to do is we're going to take a more detailed look at matching up the pressure transducer, the water level data, along with our sample timing to see if we can parse out inlet samples for the rising limb of the hydrograph and outlet samples on the receding limb of the hydrograph. I noted the fast drawdown, high filtration rate at 2 sites. So that I described in detail the the school site that it could be just because of the newer construction or just the age of the system. At the same time, if that really continues, or if that's, if that's real, that also does explain the lack of pollutant removal for that site. That water is just moving through that sand too fast for the sand to actually filter anything out. Maintenance access, in terms of reasons for concern, most of the sites we had, there is very good access for vac trucks, for visual observation, even for human entrance, for ease of maintenance. One site, however, I think would be very difficult to understand exactly what needs to occur for maintenance, and then confirming that if maintenance is necessary, was it actually conducted correctly? Was there actually enough material removed that the system can be restored? I think the last comment there, sand washing through drain tile, is some of those higher TSS concentrations that we saw on the effluent. Is there a chance that that is actually the sand, the filter media, that's flushing through the drain tile? I know there's some variation in design around that drain tile envelope itself. Some designers like the drain tile with a sock. I think the manual recommends drain tile, choker course, and then the filter media. So that is something else to look more closely at and compare some of the data on the sites. We are comfortable with a couple recommendations here. We have, in our original proposal to the research committee, we proposed this in 2 phases. Phase 2 would be for 2021. I'd like to have that conversation again with the committee to see if they are interested in studying in 2021. Based on the samples that we took this year, our stats analysis indicates that we should be targeting 25 events. Now, I'm not sure how reasonable that is over one summer if we stick with grab samples, but would be a goal for us. I'd also like to target some larger sites, 1 to 2, maybe 3 acre sites, more big box, big parking lot sort of sites. And then further refining the sample timing. It's, as you can probably imagine, with grab samples and alley being awesome, can be difficult to sample Friday nights in the dark, Saturday mornings, whenever. So it makes for difficult timing by doing grab samples. Some recommendations for designers is continuing to educate your owners, your clients, as to the limitations and the maintenance considerations. Ultimately, at some point, these systems are going to need to be, we need to restore the filtration capacity of that sand layer. With our age of our oldest system, 8 years, it doesn't look like it's going to be, that particular system has lasted 8 years. So maybe it's going to fail in 10, 15, 20. But are owners ready for that sort of investment? A couple $100,000 to rebuild this sort of system. I think a key one is to include your drain tile as an outlet device in the model calculations. Including that 4-inch pipe or whatever that drain tile size might be is critical because of that unknown filtration rate. That filtration rate in the filter media might be 1 inch, it might be 8 inches per hour. So if it's really high, if that water is just shooting through the sand layer, that 4-inch drain tile is going to act as probably the governing device in your model or your calculations. Human access at inlets, outlets. This is, through this work and then other design and review work that I've been doing, this seems to be becoming more clear in terms of where do we want access points, and do we want access points for people, or are cleanouts good enough? And I'm continuing to trend towards human access, so we can see, we can get down there, we know exactly what's going on at inlets, outlets, and at the bottom of these systems. Considering layers of pretreatment, rather than just a sump manhole, is there a way that we can do a sump manhole for big stuff, a swirl separator for a little bit smaller stuff, and then the water goes into these systems? I know we're talking about more cost, but it ultimately gets back to how are we best protecting our downstream water resources? We're spending hundreds of thousands, maybe millions of dollars on these systems, and we got to make sure that they're working. And then finally, I'm not trying to promote an underground sand filter or an MTD. I just want to point out that considering an MTD, a manufactured treatment device, instead of the sand filter, that MTD can be placed almost adjacent to your storage, so you have more easy access to that you can clean, you can refurbish the MTD more easily, perhaps more costly, I understand that, but that can be accomplished probably at less cost and easier than replacing that sand filter layer below your storage. I apologize, it looks like it is 10:43. There's only a couple minutes here for questions, but be happy to entertain those. Thank you again very much.
Great, thank you, Todd and Allie. We do have time just for a couple of questions. I will encourage you to continue to put questions in the Q&A. Even if we can't get to all of them this morning, we'll have Todd and Allie address those afterwards, and we will post those on the website with the recordings as well. So go ahead and continue to put your questions in. But we'll take a couple here, Todd and Allie. I'm going to start with the most frequent question here. Were the inlet samples collected upstream or downstream of any existing pretreatment devices?
They were collected upstream of pretreatment. As water was flowing into those pretreatment devices.
Okay, upstream of the pretreatment devices.
Yep.
Another question is, do you have data or opinions on the functionality of the geotextile in these systems? Are they good at separating the layers over time? Do they tend to block or clog water movement over time?
Great question. And it turns out that all 4 of these systems did not have geotextile separating the storage rock from the filter media. The geotextile and the liners that I mentioned, those would have been on the very bottom of the system.
Regarding your sampling, most of your samples were collected in the summer. Did you miss spring seed and flower and the fall leaf drop, or were they part of the samples collected?
I know you guys haven't heard my voice yet. Our samples were collected from mid-August to mid-October. So we did miss the spring and part of the summer, but that fall leaf drop, that would have been potentially seen in our samples.
Great. Just a comment here. It sounds like a lot of people are very interested in this presentation, this work. Thank you, Todd and Allie. A comment here is regarding more details regarding the sampling methodology and sampling timing. And so I would just add that we'll be having a mid-project report from Todd and Allie, and we'll post that on the site, and you can peer into their methodology. And we're pleased that the Minnesota Stormwater Research Council will fund this work continuing in phase 2 for 2021. So I think you all saw the limited data collection and the high interest here in this work. So we'll continue the investment to more data collection for this type of work. So unfortunately, we are all out of time for our first presentation. A virtual round of applause for Todd and Allie. And thank you for joining us. And watch for their continued work into 2021. Our second research spotlight this morning actually starts to take off from a topic we heard during our first presentation. Is you heard from Todd, and in one of the pollutants they observed and measured was having to do with E. coli and bacteria. And you might have saw those high rates at one of those sites. So our second research project we're going to hear about is actually looking at that very concern and problem, looking at bacteria and pathogens in stormwater runoff and in stormwater reuse systems. Our second presentation is going to be presented by Dr. Satoshi Ishii, and his project is looking at the temporal dynamics of pathogens and antibiotic resistant genes in raw and treated stormwater. In other words, the bacteria in stormwater runoff and going into reuse systems, which many of us know are proliferating across the state. So this work is looking at observing bacteria and pathogens in that stormwater reuse and the reuse systems. And his work is looking at 5 different stormwater reuse systems here in the Twin Cities metro area. Dr. Ishii's work is also a phased project. He's going to be talking about phase 1 of his work today, and this work is going to continue for another couple years, funded through the Stormwater Research Council. Dr. Satoshi Ishii is an associate professor here at the Department of Soil and Water and Climate, and also at the Biotechnology Institute. He has a long and distinguished career, including work in Tokyo, Japan, but also here at Iowa State and the University of Minnesota. With that, I will turn things over to him to talk about his research project. Thank you for joining us this morning, Dr. Ishii.
Thank you, John, for the kind introduction. I'm very excited to share my research funded by Minnesota Stormwater Research Council. This study was done in collaboration between University of Minnesota and Minnesota Department of Health. As you can see the names here, I am Satoshi Ishii, and the research was done with Tim Lapara in the Department of Civil, Environmental, and Geoengineering, as well as Nancy Rice and Anita Anderson at the Minnesota Department of Health. Tammy Walski and Valerie Durings are the graduate students supported by this work, and most of the work that I present here is done by these 2 students. So a little bit about the background of this research. In the past half century, there is an increasing water demand, as you can see in this slide. And most of the water is taken from the groundwater, as you can see the increasing groundwater trend. And but the groundwater is limited, so we'd better find another way to conserve our groundwater resources. One approach to conserve water resources is to reuse water. Water reuse is the use of treated or untreated rainwater, stormwater, or wastewater for various purposes, such as groundwater recharge, irrigation, car washing, toilet flushing, etc. This practice can conserve our groundwater resources, but when we reuse stormwater, it can also improve surface water quality, because nutrients in stormwater or rainwater can be removed or used for irrigations, rather than discharging into the surface water body. And the water reuse is in practice in many different areas in Minnesota. As you can see below, this is the ballpark in St. Paul. They collect rainwater nearby and treat them and use for field irrigation as well as toilet flushing. One of the major concerns about the water reuse is about the safety issues. The questions often asked is, are there any pathogens present in the reclaimed water? And what kind of reclamation processes are most effective to remove microbial contaminants. To answer these questions, we have been working on various water reuse research. One of the research was funded by LCC-MR, and this is done from 2017 to 2019. We collected water samples from various reuse facilities throughout Minnesota. And we collected 2 to 3 samples during the 2-year period. The major purpose of this research is to obtain the overview of the Minnesota reuse status and quality. Based on this LCC-MR funded project, we found that most of the reuse system uses stormwater or rainwater as the main source of reuse. The light blue pie is stormwater, and the light green is rainwater. So like 3/4 of the reuse facilities use storm or rainwater as the source. And the coarse sand filtration is commonly used as a mean to treat stormwater or rainwater. UV or chlorination is less commonly used. This brown pie is the coarse filtration, and UV is orange and chlorination is yellow. But you can see there are different treatment technologies are used to clean water. Also, we found that irrigation is the main application purpose of reuse, this gray pie. But the water is also used for toilet flushing or vehicle wash. So in this project, we collected water samples from mainly from Minneapolis-St. Paul metro areas, but we also collected from Duluth and Mankato and St. Cloud areas. So based on this research, so we also detected some potential pathogens, but since we only collected 2 to 3 samples per site during the 2-year period, the sampling intensity is not enough. That's why we applied research proposal to MSRC, and they kindly supported our research. In the MSRC phase 1 research, we focused on stormwater reuse because most of the water reuse facilities in Minnesota uses stormwater or rainwater. And we intended to collect more samples more intensively during the reuse season. And the main purpose of this research is to analyze temporal dynamics of pathogens and antibiotic resistance gene concentrations in the water reuse facilities. Okay, the objectives of MSRC phase 1 project is to monitor levels of pathogens and antibiotic resistance genes in stormwater samples, evaluate effectivity of water reclamation systems, and to analyze correlations between pathogens, ARG levels, and environmental parameters. By doing this research, we'd like to contribute to the establishment of safe and efficient reuse of Minnesota's stormwater. We collected stormwater samples from 5 locations with different treatment and application purposes. These facilities are located in 5 counties as shown in this table. They are all located in Minneapolis-St. Paul metro areas. One site in Carver County uses coarse sand filtration and chlorination to treat their stormwater. their application is to irrigate in the lawn. One site in Hennepin County do not use filtration. They have a large stormwater pond and a screen to filter out like leaves, but there's no there's no sand filtration or disinfection at this site. And again, they use irrigation for their stormwater. A site in Ramsey County uses multiple layers of filtrations and disinfections. They use vortex bag filtration or also charcoal filtrations, as well as UV disinfection. The main purpose of this site is toilet flushing. A site in Scott County, they use coarse sand filtration, but without any disinfection. And site in Washington County uses coarse sand filtration together with UV disinfection. Both of these sites use their water for field irrigation. We collected 3 samples per site before and after treatment and at application site. We collected samples every 2 to 3 weeks from June to October in 2019. So the total number of samples are 99. In addition to stormwater samples, we collected city water samples as a control. These samples are collected at the field irrigation sites. After water sample collection, we capture bacterial and virus cells in a membrane filter and brought these membrane back to the lab. In the lab, we recover bacterial cells and viral particles and extracted DNA and RNA from these cells. The DNAs and RNAs are used for high-throughput multi-pathogen quantification. This is a microfluidic device to quantify many different kinds of pathogens for many samples. At the same time, we also collected water for chemical analysis, such as temperature, turbidity, and chlorine concentrations. And also we counted E. coli in the water. The target pathogens and antibiotic resistance genes include E. coli, Salmonella, Campylobacter, Legionella, etc. There are a total of 23 targets. For viruses, we targeted waterborne human pathogens such as norovirus, adenovirus, etc. In addition to pathogens, we quantified antibiotic resistance genes, 47 targets. ARGs are the emerging contaminant of concern. So that's why we included these in this study. So this is a summary of pathogen and ARG results. From 15 to 35 different ARGs pathogen genes were detected. ARGs were more frequently detected than pathogenic bacteria and viruses. Among the pathogenic bacteria and viruses detected, enteropathogenic E. coli and human adenoviruses were relatively frequently detected across samples. This figure shows the average concentrations of genes detected across 5 different sites, as well as tap water control. All sites have similar gene concentration, even from the tap water control. The average concentration is relatively lower than the stormwater reuse sites. But this concentration is relatively comparable at the Ramsey County site. This is probably because the reuse facilities at the Ramsey County sites use multiple layers of filtration and disinfection. We have collected lots of data about pathogens and antibiotic resistance genes. It is a little difficult to show the overview of the results at once, but I broke down the results at each site, and I will present 2 sites in this presentation. One site is at Carver County site. So this figure is a heat map, and darker color, darker cells means the concentration of these genes are higher than lighter cells. So for example, these brown cells, these are actually genes for Salmonella. MBA and TTRC gene, these gene encodes for Salmonella. So this means Salmonellas were detected at high concentration at this date, September 4th. Similarly, this gene, EAA, this is from enteropathogenic E. coli. This gene is detected at relatively high concentration on June 5th. You may also see that the clustering of samples by gene detection patterns. So these samples, they are all collected on July 2nd. They are clustered together. That means their gene profiles are similar to each other. What that means is in these samples, similar genes were detected at similar concentrations. You can visually see that based on this heat map. For example, you see these genes are detected all in these 3 samples. One interesting thing at this site is the samples, there is a big storm event right before the correction on July 2nd. So this is a precipitation date on the top right corner. There is a precipitation at 48 millimeter and 52 millimeter, one day or several days before the sampling event. This is about 2 inches of rainfall. And this storm may have influenced the turbidity. As you can see in this turbidity plot, the turbidity increased on the date on July 2nd. And this is probably related to the storm event. There are 3 lines, orange, blue, and gray. And orange and blues are, I think they are collected before treatment. So, and this is the after treatment. So that means their treatment system can reduce turbidity at some extent. However, we didn't see any increase of E. coli on July 2nd. Their concentration is relatively small throughout the sampling season. There is some increase, but this increase is not so big. It is still below 30 mpn per 100 milliliter, which is relatively low. Another example to show is at Scott County site. We also detected enteropathogenic E. coli and some other pathogens. But again, we see that the occurrence of pathogens are rather sporadic. Yeah, this is same for the Cover County sites. We detected Salmonella and enteropathogenic E. coli, but this is just once a year or so, not consistent throughout the sampling season. In contrast to the Cover County site, we didn't see tight clustering of samples based on the pathogen and ARG concentration profiles. We see some precipitation in August 20th, and we see the increase in turbidity. But that precipitation didn't influence much for the gene concentration for E. coli, gene concentration for pathogens and antibiotic resistance genes. So August 20, so we collected samples right after precipitation event, which is August 22nd. But these, we didn't see obvious increase in pathogen genes right after this spring event. However, we observed the increase of E. coli after the precipitation event. So this is in contrast to the Cover County sites. So maybe the trend is depend on the site or reuse systems. We also did some correlation analysis between total number of genes detected and some of the chemical parameters we obtained. So this total gene copy means this is the number of genes detected in the samples. There is some levels of correlation with E. coli and total coliform levels, but the R square, the correlation is not so large. We may need to break down the total number of genes to further find out the correlation between environmental parameters and like concentration of genes for specific target. So the conclusion of our phase 1 research is that the precipitation could influence stability and occurrence of E. coli and ARGs, but this trend is site or system dependent. This may be related to our sampling method. We collected samples when the system is on, because we went there usually in the morning or at the daytime. And the water reuse, they apply the water for land irrigation, usually at the very early in the morning. So what we did was we collected stagnant water from the system. And we collected water samples not always after a storm event. So that is the limitation of the phase 1 research. We also found that the water quality at Ramsey County site was comparable to tap water control. But this does not necessarily mean that other sites did not do a good job. The water quality needed are dependent on application purposes. We may not necessarily have to have the tap water quality water to irrigate or to flush toilet. So in order to assess or evaluate their systems, we may need to do some risk assessment and evaluate how clean is clean or how clean is enough for your purpose. So this is the phase 1 research. So based on the results of phase 1 research, we found that we need to collect water when the system is on. So thanks to the MSRC, our next proposal, our phase 2 proposal was approved, and we collected samples in 2020 season. We are currently analyzing these data to obtain more solid conclusion. Before closing my presentation, I'd like to share some of the outreach activities we did. We did outreach event at the Minneapolis Northeast Open Streets. We did a poster presentation to explain why stormwater reuse is important to conserve groundwater resources and to improve surface water quality. We also did a PET bottle sand and charcoal filtration device to see how we can clean up water. We also handed out MSRC leaflets to the public. So I hope MSRC likes this event. With that, I'd like to thank the students who helped collect samples for this event, and also many thanks to the stormwater reuse facilities for allowing us to collect water samples from their system. This research was supported by Minnesota Stormwater Council as well as LCCMR. With that, I'm happy to answer any questions you have.
Great. Well, thank you, Satoshi. Some great information. And as you all heard, we are working continuation of this project into 2021. We do have some questions and we have some time for those. Continue to enter your questions and we'll get to as many as we can here by 11:25. Satoshi, so based off what you've seen so far, do your results indicate a pathogenic risk from reused stormwater, you know, and using this on sites? What's our risk, or is there?
Okay, it is a little difficult to answer with our current data. We detected some potential human pathogens like Salmonella and Enteropathogenic E. coli. In order to assess risk for risk to public health, we need to do quantitative microbial risk assessment. And to do that, the number of detects in our samples is not enough. So that means we need to collect more samples to have better information for the risk assessment. But with the current data, the number of detects is not so many. So that means the risk associated with the stormwater in these sites are not very high. Like, I may say that.
Thank you. So based off what we're seeing, what you reported, it does appear that relatively little treated stormwater, let's say from stormwater ponds, would that be suitable or dangerous for irrigation uses and spray? I know that's different than what you've studied, but based off what you're seeing as pathogens and bacteria and reuse systems related to stormwater runoff, what's the risk or usability of stormwater from these other practices in your opinion?
That's also dependent on the source or the, how stormwater is collected. Is it from the parking lot or is it include some, are there any potential contamination from dogs or geese? That these, dogs and geese may be the source of pathogens and they may pose some risks for contamination. So it depends on the site, but based on, we collected water samples from different sites and we didn't see any big difference between coarse sand filtration treated water and no treatment water. So based on our limited data, we don't, at least I personally do not see big risk of using untreated stormwater, but we don't know yet. We need more data to
Great. Say, I'll ask a question here. Since you mentioned geese, does this type of work in your work able to differentiate, I guess in regards to like E. coli, the source, geese, pet waste, dogs, does this type of work help us better understand that, Satoshi?
Yes, there is a method to differentiate sources of fecal contamination, which is called microbial source tracking. And by using that approach, we can determine the relative contribution of, for example, geese and dogs. So by doing that, by clarifying the source of contamination, we can do better management practices.
So another question here is, what is considered good water quality for different uses? I guess maybe if you could talk, and you mentioned irrigation. So based off what you saw, did these sources meet the criteria for irrigation?
Yeah, that is a very good question and very difficult to answer. So in order to answer that question, is we need to do some risk assessment. So the potential risks for like drinking and toilet flushing and irrigations are different. So I mean the water quality requirements depends on the application purposes. And to clarify the acceptable concentrations, I would say, we will need to do risk assessment. And we have not done that risk assessment in detail yet. We are planning to do that, but we don't have the detailed information yet.
All right, let's take one more question here. It looks like some of your sampling events showed low E. coli, but pathogens were identified. Was the relative level of pathogens found an issue? Could you talk a little bit more about the pathogens that you detected?
So E. coli have been used as an indicator of fecal contamination, and that is based on the assumption that E. coli are coming from recent fecal contamination. But some pathogens may also survive in soils, and they naturally occur in the environment. So E. coli does not necessarily correlate with the occurrence of pathogens. So it is not surprising to me to see the detection of pathogens, even though the E. coli level is low.
Great. So one final question here is, so the temporal dynamics are mainly concerned with time from the precipitation events. Is that true?
Our purpose of the phase 1 is to capture the impact of precipitation on the pathogen concentration in water. But we also interested in seeing the seasonal dynamics. So for example, if temperature rises, pathogen concentration decrease or increase. So both. We are interested in detecting stormwater impacts as well as long-term seasonal trend.
Great. Well, very informative, and thank you very much, Satoshi, for presenting your work. And I encourage all of you to stay tuned as he and his team unfolds the work in phase 2 here in 2021. With that, we're going to wrap up today's seminar. I would encourage all of you to stay informed about our upcoming agenda for seminars, which is located on our website. And look forward to joining us on January 21st. We'll announce registration for that the first week of January. So please join us for that. Remember that all our past seminars are recorded and available online, and you can find them and view them at your discretion. So with that, thank you all for joining us. I hope you find these seminars informative and useful for your professional development and your work in stormwater. We wish you all a happy holiday season and look forward to working with you in 2021. Have a great day, everyone.